Before I start, I just want to acknowledge a couple of research students who have driven elements of this work, and also Andrew Baird, who's pretty close to a 50-50 collaborator on one of the other elements. For, well over a century, one of the major questions that's been addressed by community ecology has concerned how large numbers of species can coexist when they compete for one or a few shared limiting resources. And nowhere is this problem more salient than in the tropics, because of course coral reefs and rainforests are the most species-rich ecosystems in the world. What I want to do today is talk about the effect of connectivity, that is patterns of dispersal between patches of habitat, on this question about what explains biodiversity maintenance. Of course, it's also important from a management standpoint because of the increasing focus on spatially structured management through things like no-take marine reserves and so forth. But what I want to do is talk about a little bit of work addressing the importance of connectivity for understanding biodiversity, and particularly the effect of complex dispersal patterns on the ability of species that compete for space to coexist in an ecosystem. And then I'm going to move on to talk about a little bit of work related to some of the challenges associated with estimating what these complex dispersal patterns are. And I'll talk both about some work on the physical side and also some on biological impacts on dispersal. So before I talk about modeling connectivity and biodiversity, I want to introduce you to two key concepts if you're not familiar with them. One is self-recruitment and the other one is spatial asymmetry. So the best way to understand self-recruitment, imagine a sort of idealized world where there are only two patches of habitat, say Reef A and Reef B, and the adults in that habitat produce offspring like larvae that either come back to Reef A or they go to Reef B or they don't make it back anywhere. So self-recruitment is essentially the percentage of offspring produced on in one of these habitats, say Reef A, that come back to Reef A of the total that are ultimately successful. So if you have 80% self-recruitment, 80% of the ultimately successful propagules come back to Reef A, the other 20% must disperse to Reef B. Same thing is true for Reef B. So any point along this red diagonal here is what I would call symmetric dispersal. That is the degree of self-recruitment to patch A, the degree of self-recruitment to patch B are the same. So there may be, self-recruitment might be low, might be high, but it's the same for both patches. Now, as we move up and to the left on this graph, perpendicular to that red line, dispersal becomes increasingly spatially asymmetric towards Reef B. So most of the ultimately successful offspring from Reef A end up on Reef B. Most of the offspring that are ultimately successful produced on Reef B end up on Reef B. Diagonally down and to the right, we get increasing asymmetry towards Reef A. Now, this is important because existing theory, that is models to explain the coexistence of effects of connectivity on species coexistence, consider one of two special cases. One is symmetric dispersal, where both species dispersal patterns fall on that red diagonal line. The other one is if they do allow for spatial asymmetries, which you can think of as source-sink type dynamics, they assume that the interacting species have essentially the same source-sink patterns. The reason that's important is because, of course, in nature, species differ in their dispersal patterns. They disperse at different times through different mechanisms. They have different characteristics that make them respond in different ways to things like, for example, ocean currents. And also, processes that push organisms around tend to be complex. So ocean currents, we had eddies and currents going in different directions, and so purely symmetric dispersal probably almost never happens in nature. So we might ask the question, is this important? That is, if we allow for these additional complexities, does our understanding of the effect of connectivity on biodiversity change, or is it basically the same? So to try to address that question, Yakov Solomon, as part of his honors, put together some models for the dynamics of a bunch of species living on a bunch of different habitat patches like reefs that interact with each other by dispersing their offspring between the patches and competing for space, for access to space in the habitat. So the model is actually a system of equations for the changes of abundance of all the species on all the patches. It's at the end of the talk if you want me to show it to you, but what I'm going to do now is just diagram the important bits for this talk. So again, think about the idealized universe where we have only two patches, patch A and patch B. We have adults that live in these patches, and they die through things like disease and predation and other sources of mortality. They also produce offspring, and of the offspring that are ultimately successful, some fraction of them come back to where they were born, and that fraction might be different on the two patches because we want to allow for spatial asymmetry, and the remainder of the successful ones then have obviously dispersed to the other patch. Now, this would be one species. It's interacting with other species, competing for space, and so those other species also have the same sort of demographic patterns, but the pattern of the level of self-recruitment or the spatial asymmetry might be different because we want to allow for these species differences. So what I'll do now is essentially show you some of the results, a couple of graphs of results that illustrate the key concepts. For this model analysis, we have two species, a competitive dominant, which has a birth or survival advantage, and a competitive subordinate. So again, this is self-recruitment of the dominant to patch A and patch B, and I'll first consider the case where the subordinate, competitive subordinates dispersal is strictly symmetric. Now remember, on this diagonal red line, dispersal patterns are spatially symmetric for the competitive dominant. And if the dominant's dispersal is spatially symmetric or close to it, the subordinate can't stick around. It's competitively excluded by the dominant in keeping with existing competition theory. But if we allow for spatial asymmetries in dispersal, then it becomes easier and easier for the competitive subordinate to persist. And so we end up with both species able to coexist. If we consider another case where the subordinate disperses asymmetrically, in this case to patch A, again, if the dominant disperses close to symmetrically, it competitively excludes the subordinate in keeping with existing theory. If we allow for asymmetric dispersal of the dominant to patch B, then coexistence is easier. That kind of makes sense. The dispersal of offspring is disproportionate to different patches. But somewhat surprisingly, as the dominant disperses asymmetrically to the same patch as the subordinate, that also makes coexistence easier. Unless the dominant disperses really asymmetrically, then it can actually be competitively excluded. So there's a host of these kinds of model analyses, and the bottom line is that asymmetric dispersal patterns, when they vary between species, promote the maintenance of biodiversity, promote coexistence of competitors. That gets easier and easier and easier the more patches there are in the habitat. And this is even if there's no environmental variability among the patches. So there's no niche partitioning in this model. This result also appears to be robust to different ways in which we can characterize competition for space. And what it tells us is that dispersal mediates the maintenance of biodiversity much more powerfully than previously thought. So what does all this mean? Well, this is a theoretical model, and so essentially what it tells us is the universe of possibility. Another way of saying that is it tells us if what we want to explain is biodiversity maintenance, it says, well, one of the things we should be looking for is spatial asymmetries in dispersal. We should be trying to work out how strong they are, what their nature is, and so forth. But if we want to ask how important this mechanism is in a particular system, like coral reefs, then we need estimates of dispersal for that system, and that's essentially empirical. And so what I want to now do is briefly talk about two challenges in the estimation of dispersal on coral reef systems. One is on the physical side, and that deals with estimating the retention of larvae in the wake of reefs by fine-scale circulation features. And the second, on the biological side, deals with quantifying the patterns of acquisition and loss of the capacity for corals to establish in the adult habitat, what I'll call settlement competence. So first I'll talk about the physical side. One of the big problems with understanding how ocean currents push larvae around is that connectivity, dispersal between reefs, can occur over really big scale. So this is a hydrodynamic dispersal model from Rob Cowan and colleagues for the Caribbean. These blue lines indicate, well, all of the lines indicate sort of demographically significant levels of exchange of individuals between reefs. And this significant exchange of individuals can occur over hundreds of kilometers. At the same time, the transport of larvae is affected by extremely small-scale features illustrated here. So the blue blob is a reef. The arrows represent what ocean currents are doing. You can see there's this quiescent area behind the reef where an eddy is formed in the wake of the reefs. And we know from plankton toads and so forth that larvae accumulate in these wakes, and they can, in some cases, stick around for several days. The reason this is a problem is, of course, to calibrate hydrodynamic models that accurately represent these features and their effects on larvae requires a lot of instruments, which are extremely expensive and requires a lot of time. Also, to model these kinds of fine-scale features on this kind of a scale, tens to hundreds of thousands of square kilometers, is computationally incredibly expensive. And so effectively, characterizing this kind of a pattern for all of the reefs in a huge region is impossible. And so one of the things that we're trying to do in the lab, this is Paulina's PhD, is coming up with ways of approximating local retention that don't require explicitly modeling these fine-scale 3D hydrodynamics. And so this graph here shows there's a bunch of points on this graph. Each point is the result of a fine-scale three-dimensional simulation model of larval retention. And so it shows how long larvae are retained in the wake of one of these reefs. The reefs in these simulations are different shapes, kidney-shaped, triangular-shaped, and so forth, different sizes. There are different current speeds, different levels of tidal fluctuation, and so forth. The bottom line is that the variation in retention time, which is about an order of magnitude from less than half a day to almost a week, about 90% of that variation is explained by a non-dimensional retention index, which is essentially a function of the average flow speed approaching the reef, the width of the reef, and the height. So three things that are actually really easy to measure. And a power law relationship explains about 90% of the retention time that's actually predicted by these complex models. So suggesting that we have a nice quick and dirty way of getting a pretty good approximation of retention times. Okay, so I'm going to move on to the biology now. Of course, biological processes also affect dispersal, behavior, survival, all that kind of stuff. And the modeling of these processes in dispersal or connectivity models is often very highly idealized. And one of the things that's very highly idealized is settlement competence, the capacity for a larva to successfully establish in an adult habitat. A typical assumption is shown here. It's what I'll call the rectangular competence model. The idea is larvae spend some period of time in the plankton developing and becoming capable of settlement. Then at some time they all become able to settle, and they retain that for some for some maximum planktonic duration, and then they all lose the capacity to settle. The problem is that data suggests quite a different pattern. This is the coral Acropora valida, and you can see that competence is lost quite gradually in a cohort of larvae from about 3 weeks after spawning all the way out to about 4 months. And so what Andrew and I wanted to do was put together some new models for the competence, also the survival, which I'm not going to talk about, dynamics of coral larvae. We wanted to be more realistic than the rectangular model, but we wanted a model that was relatively simple, not a lot of parameters, so that it could be calibrated from the kind of data that we might realistically actually be able to collect, right? We can make really, really complex models, but if the data don't support the model, then it's kind of a glorified computer game. So what we did was calibrate the model for 5 species of scleractinian coral, 3 branching acroporas, 2 slow-growing massives, some ongoing work with other species. But essentially, again, the model is a bunch of equations for the dynamics of larvae transitioning between different states. Again, I won't show you the equations, but basically, these are the states larvae can be in. They can be pre-competent, not yet able to settle and metamorphose into juveniles, or they're still alive, but they've lost the capacity to settle. And so larvae transition between these states according to acquisition and loss rates for competence, and the larvae are also subject to mortality. The sort of a competence pattern that this model predicts is this sort of a thing. So competence switches on, there may be a time lag, then there's a peak in competence, and then there's an exponential-like tailing off in competence. Now, if we calibrate this model to empirical data on settlement competence and culture, what we get is these patterns here, particularly for the species where I have a reasonably well-resolved tail. You can see there is a kind of exponential-like tailing off in competence. If we use standardized transformed residuals and various fancy ways of checking model fit, in fact, we find the model works pretty well when we consider its fit to all these different species in the aggregate. And the key point is that competence is, in fact, lost quite gradually, and the model picks that up reasonably well, certainly better than a rectangular competence model. we can combine this with calibrated models of larval survival and produce what I would call a temporal dispersal potential. This is the proportion of an initial cohort of larvae that would be alive and capable of settling as a function of time. And so essentially what we can do is use this kind of a calibrated model instead of those idealized rectangular competence models and so forth in oceanographic models for dispersal, right? So the idea is that now in an oceanographic model, you put larvae into a parcel of water and the larvae get pushed around. When they're close to a reef, you work out how many of those larvae are able to settle into the adult habitat. We can use the calibrated model instead of some of the cruder rectangular competence models, and also avoiding assumptions about constant mortality rates and so forth, which I didn't have time to discuss. So basically, I want you to take two messages from this talk. One is that complex patterns of dispersal have quite large and important effects on biodiversity, and in particular, the maintenance of biodiversity or the coexistence of species that compete for space is a lot more likely when there are spatial asymmetries in dispersal that differ among species, and this is not something that was accounted for in competition theory really before this year. And the second thing is that quantifying these complexities to ask how important this mechanism is in a particular system like coral reefs in tractable ways that are broadly applicable that we could use in a lot of different regions of the world is a major challenge, because a lot of the associated with a lot of these logistical problems with things like calibrating 3D circulation models. But I hope I've convinced you that some quite simple models can actually provide very good approximations for both the physical and the biological processes that influence dispersal in the coral reef environment. So that's all. Thanks, guys.